Hello, everybody. Here we are, finally. So, sorry for the wait. Had a lot of unforeseen little difficulties kind of crop up, so sorry about that. We are here with Scooby, and to start off, to allow a few more people to find us, we're going to go ahead and listen to a little bit of Stacy. It's one of my favorites. Here we go.
Yay! I don't know if I can see the comments here.
A few more people to find us. We're going to go ahead and listen to a little bit of Stacy. It's one of my favorites. You need to mute something, Scooby.
Yeah. I don't know if I can see the comments here. Okay.
Yeah, let's keep talking till he's.
All right, we're going to back that up.
So let's keep talking about Avery.
About Avery. Oh, I gotta get the tambourine.
Let's keep talking till he's set free. Oh, you just listen, I think
Hey, everyone.
why it matters.
Matters.
Really matters.
Really matters.
Put aside the documentary, the state's version of mediocrity. Oh, all their claims so blatantly on the tatters.
It matters.
I think that matters.
It matters.
I bet you heard about her last sweat. Maybe you ain't heard this yet. 19 more times the DNA that was left anywhere.
Oh, my wires are all taken.
I think that matters.
I think it matters.
I really do. I think it matters.
I think it matters.
An ex-boyfriend with a day planner page, and yet they did not investigate. And who knows why, and who can say why you were signed into the crime scene that day.
I think it matters.
I really do. I think it matters.
Oh, yeah.
About the barrel that one was
seen. Yeah, those barrels.
To the Avery. The bones were found. The next day was not one photographic disc of where they lay. I think that matters.
Oh, yeah.
I really do. I think it matters. I could go on listing things off. 80 more things. I just keep on singing these songs. To every freedom rings, I think it matters. It really, I know it matters.
It matters.
It could be you, I think it matters. It could be me, I know it matters.
I think it matters.
Work with Casey, it
matters.
I said it matters. It really matters.
All right, folks.
Hi, everyone.
We are trying to get Stacy to 1,000 subscribers, and so that he will be able to go live from Raleigh this year. So, trying to get Stacy to 1,000 subscribers. Remember to go over and do that. All right, we're here today.
All your friends.
Sorry.
Tell all your friends, Stacy. I'm advertising.
Yeah. So.
Yes. I'm
getting my today talking about obviously
Papa Dr. Pepper, Erekose. Get your second wind.
So we're here today talking about the 85 blood vial, the discrepancies between which is where the sample in 96 was taken from, and the discrepancies between the results there and the results in 2006. And obviously, you guys know we were on with Razor a few nights back. I believe that was on Friday night. So we're now back with Scooby, and we're going to hear a little bit of what Scooby has to say about some of this, and then we'll obviously we're going to be kind of doing a kind of a flyby on this right now, maybe an hour, and we're going to probably come back in a little bit more detail at a later date. So just to warn everybody.
Jason, that was the worst advertisement ever. Well,
Yeah, James has been making some rather
oh, James. Oh, okay. Well, James.
So, but that's all right.
John Smith, message deleted. Oh, perhaps the trolls are out tonight. Well,
anyway, so
we welcome everyone.
We're here to talk about tonight, folks. Is not
Great.
that. So, let me just show you what we are here to talk about.
I wonder.
Okay, so you guys remember I showed you guys this when I was talking to Razor and we were going over this and stuff. Well, this has a little bit added to it. Just a little bit added, substance here. So, obviously, you guys remember when Razor and I were talking about this. This is the alleles, or I'm sorry, the loci they tested in 1996 when they were testing the fingernail scrapings. It was these three loci, and these are the results. Now, it has been said that the fact that there's a 9 and 11 here, the 9 could very easily have been interpreted as a 9, even though it's actually a 9.3, as we see over here. Scooby said that, and even Razor admitted that did seem very possible. So that could explain that. Obviously, the I'm sorry, I'm mixed up. Okay, the 10. There's a difference between a 10, sorry, on the TH01, and then a 9.3. So there's the discrepancies here, but for me, the really, really bad one is this 7 here. And apparently, according to Scooby, it's been known to happen, and she'll explain that, I'm sure. But as we look through all the Averys and Dassies here on those loci, particularly the CSF 1PO, right? It's 12s. So it's, mostly 12s, a few little difference, but no 7s. Nowhere in there is there any 7s. And so it's very odd that we would see this 7 here. So that's, to me, a very glaring discrepancy. You're talking about the difference between, a difference in 5 tandem repeats when it comes down to it. And it's going to be interesting to hear how that mistake potentially happens. So, what do you think, Scoobs?
Okay. Well, let's go. First of all, thanks, Rexnor Rainer. I don't know how to say his name.
Razor.
Hi, everyone.
Razor.
Razor. Hi, everyone. And sorry, James. Who is it? James is a little discontent. But you know what? In a perfect world, I would be very organized and have a beautiful presentation, and everything would flow as it should. But you know what? Life happens, and so that's just always. Life is not always perfect. So let's. Nor is DNA testing. So I'm just going to talk about scientifically, Eric, if you want to go to my screen. So I just put up basically, it was just another presentation of what you did. My first thought was obviously in 1996, the blood was tested by a private lab called LabCorp.
LabCorp, yeah.
Just what I did, I guess I'll, because this required going back in time, when Erekose and I were talking about this, I guess we're not going to go into this in full detail tonight, but my first thought was it's a testing error. You're correct in that obviously it makes no, the 7 and a 12, that's a big difference. However, I wasn't sure exactly what the other lab did. So I actually did write to someone who is, I think, one of the leading experts in the country. And he did write back to me. And so I'm going to share that email with you. Now, barring some type of foul play, as in this was not his blood, the most likely explanation is error in testing, especially when I explained to you the difference between the testing that was done in 96 and the testing that was done later. I'm not trying to convince anybody one way or another. I'm just trying to explain some of the scientific rationale, and if you understand the testing. And also, and this is probably what we'll get into more because we won't have time tonight, the bigger picture that errors occur. Errors occur. This was old testing. And some of the, I'll go over a little bit, like you said, the 9.3 issue, kind of makes sense with the older testing not being as discriminatory. But errors occur all of the time in the forensic, all kinds of errors. And we can talk about, many different kinds of errors. But
Ziggy's asking if you can enlarge that. Can you hit the zoom on that and zoom in on it just a tad?
Yeah, it's taking in my whole screen. This is just
no. Just go up. There's a little thing right there with a magnifying glass, right? Go to that.
Oh yeah. Oh yeah. Okay.
And then slide that bar over a little bit, and it'll make it bigger.
This is just is just a PowerPoint. So is that better?
That's better. Yes. Thank you.
Okay. On my screen, it looks so big. Okay. So this is just basically another representation of what Erekose showed you. So this is just a summary. So this is Stephen Avery's complete STR. STR stands for what, people?
Standard repeat.
Short tandem. I'm going to make you all think. We're going to have Scooby Skull. Short tandem repeat. So short tandem repeat. Hi, Kim. So this is the Stephen Avery's full profile that was done by Sherry Culhane, and probably the one that was done in 2002, which led to his exoneration from the first trial. This is what would have been entered into the Wisconsin state database, and this is what would have been pulled out of the state database when he was for his later conviction. Now, as you see, there's all of these loci that are tested. Back in 1996, this was done with a prefabricated testing kit, which is commonly how these tests are done now. So you can look up, and we'll go into this later, exactly what test was used, and we're going to talk about primers and how, and some of the things that cause errors and tests to be different. And I'm even going to show you that there was a study done recently where they actually, for a long time, the DNA evidence was always considered infallible. There weren't any problems. But now, more and more, and of course, there's a lot of resistance in the forensic world to exposing certain things. But now we know more and more that that's not the case and that errors happen. So in 1996, this is the basic lowdown. When his blood was tested, and I'm going to talk about the differences between the testing, they didn't test all of these loci. So it's not that these things didn't show up. It's just that these were the only 3 that they tested for.
Right. And it was explained that Stephen's defense team they actually stipulated that those were the 3 loci that they were going to test. It was actually, I guess, his appellate attorneys who determined that.
Yeah, because there was only nowadays this is all automated, but back then you had to do it by hand, one at a base, so to speak, or manually, one at a time. And so here they got a 9.3 and a 10. We'll talk about it. This is the problem one, the CSF. These are the loci, the locations on the chromosomes that were tested. This is the one that's, of course, the big deal. and the TPOXX is the same. So, not wanting to get you guys good information, I actually wrote to someone who I do consider a uber expert in this area, Dr. Dan Crane, and I'll give you links to all of this information. He has, by the way, at least 10 videos on YouTube, which I think are excellent. In fact, I'll put them in my Scooby School, which I'll get going. He actually comes from the academic world, which is always, sometimes you guys hear me rip about the forensics world, but he's a professor of biology with a PhD in molecular biology, which is sort of DNA. And but he also has been kind of in the forensics world since way back, and he actually operates a company and a website called Bioinformatics, and I'll give you the link to that. It has a lot of educational stuff. So he actually works with lawyers and works with the legal system. Anyway, so I think he knows this stuff, and I thought he would understand what was going on back then. So he sent me an email. Can you guys see this on the screen? I'm screen sharing as far as I know.
Yes.
So I thought I'd show you because some of the ducks. Okay. So I basically wrote, now 2 things that I did. I explained the situation. I didn't give him any other information. A, because I didn't want him to be biased. And B, because if you mention this case, a high profile case, to somebody who's a big name, they're not going to want to comment on it. So I just explained the situation. Mm-hmm. So, it says, hi, thanks for your email. I think the simplest and most likely explanation for the discrepancies you are seeing between the 2005 and 1996 testing is error. So basically he's confirming, what I said. However, he does go on to explain, which was helpful.
Confirming how unreliable DNA is. Right. Go ahead.
Anyway,
sorry.
You mentioned, that's okay. I'm just going to read this because I'm explaining, well, not unreliable, but not infallible. And I'm going to show you.
Tomato.
It's one, if somebody, so therefore, if you are convicted of a crime that you know you didn't do, and someone says, we have DNA evidence against you, people used to plead guilty because of that. But
I do have more to say about this, but I'll let you get to the email first.
Okay. You mentioned the test kit that was used in 2005, Pro Mega Powerplex, and that's important, and this is changing too, because, now up until recently, we would know, like, for instance, we know the kit that the Wisconsin State Crime, this is such exciting stuff, we're going to have to do it. Schedule another date, and I've got so much good stuff for you guys, but anyway. So we know what kit Sherry used, Pro Mega Power Cooks, but not the one that was used in 96. That you only have results for 3 loci from 96 suggests to me that those were the only loci that were tested.
That's true.
So that's
correct.
That would have been close to the end of the era when then don't worry about these names, DQ alpha and polymarker testing, reverse dot blot tests would have been done, and close to the beginning of when STR loci would have been tested. Okay, so this is like, and that's true. 96 was about the time when some of you have heard of restriction fragment length polymorphism testing. That was the old kind of DNA testing that they used to do. And these other things that he's mentioning were some sort of interim types of tests that they did while they were switching over to this new STR testing that used PCR. Anyway, so that's what he's talking about. But those 3 loci would have also been some of the original ones that the RFLP testing would have done on as well. So what they started to do, and I'm not even going to go into explaining this older technology because it's not used anymore, but just understand that there was an older technology that was different. However, there was a little bit of overlap in that they started testing using the old technology some of these loci that you're now familiar with from newer STR testings, but they did them on a gel, on an agarose gel using autoradiography, which might not mean a lot to you, which is different than the capillary gel electrophoresis.
So the gel can make the 9.3 look like a 10. Okay, all right.
You're tired and
I'm
crabby.
fine. No, I'm just saying I can follow along with that. I
Yeah.
get that.
Well, yeah, but I'm going to show. Yeah, you're right. It's a whole different testing type of. So anyway, so I'm just trying to explain this hopefully so people understand. So I think you might be seeing results that were generated with an acrylamide gel, one locus at a time. Okay. So what I'm saying is that this type of testing was very different from the later STR testing that Sherry Culhane did and everybody else has done using something like a PowerPlex kit. Okay, so the results would have been on an X-ray film autoradiogram for each locus versus being fluorescently tagged. So the testing is very different and not as discriminative.
Yeah, right.
While it would be pretty easy to distinguish between a faux 9.3 and 10 allele with capillary electrophoresis, it would have been a bit harder with a gel, to the point that it actually took a while before the 9.3 was even recognized as being a fairly common allele. And I'll show you some charts of the alleles. So remember these, and still now new variants of alleles are being discovered. Okay, so these were not always, especially these 9.3. And I'm sure, does anybody out there? I'm not really watching the comments, but does anybody?
Carol,
I don't know if
beating me to my thunder. She's stealing my thunder right now.
People don't know do people know what 9.3 means? Probably not, but you will. Okay, because that's important too. Because if you know what nine, well, I'll just tell you before I get to my picture. So what a 9.3 means is that these are four. Oh, let me get my paper out. These are 4 repeats, right? So you got. I don't know I can't remember what they all are. Okay. Okay, so that's like 1 repeat of 4.
Right.
So if you've got a 0.3, okay, that means that you only have 3 of the 4. So that's what a 9.3 means.
Right.
So there's only a 1 nucleotide.
So it's actually more like a 9.75, really.
Exactly. So there's only a 1 base pair difference between those two. So
which is why it could be mistaken as a 10. Right. I get it.
The way that an agarose gel is done, it's not as, to be able to distinguish one nucleotide, wasn't as sensitive. So that one does make sense.
Yep.
Okay. Now, as you can see, blah, blah, blah. Okay. So there would be no good explanation why a 7 allele might later appear to be a 12 allele at CSF.
Gotta say I agree with them.
Well, right. No, we all agree. Okay. So then what? There's no good reason. Okay. But then you gotta, okay. So then why? I would expect, however, I am going to show you though that there is another one. Okay. It's very rare, but there is another documented case of this happening in the literature, but there's no good reason. Right. I mean, he's agreeing. Right. I would expect, however, so now you got to look for, I would expect that the 1996 results would look obviously bad if you saw them. Lots of noise and something that would be very difficult to interpret. So then that would be, the only reason. Now the problem, of course, is that, and I have not written to LabCorp yet, but I'm pretty sure that they would not. This is the problem in forensics and in the legal system, is you don't have access to the data. So we don't know what this gel looked like. Maybe it looked like a big hot mess and you really couldn't tell. So they just said, oh, it looks like this. Or maybe it didn't. We don't know that. Anyway, then he goes on. I was like brainstorming and trying to think of like any other possibilities that might, that I didn't think were very likely for this being different. And he agreed that those were not, basically in the last paragraph, that those were not, not likely problems. In other words, different genotypes in blood versus skin cells. While that has been reported, it's a very, very rare phenomenon. and
Right.
a somatic mutation. Again, these would be very rare things. That would be highly unlikely, and especially now that we see the later test, and see that his parents, and it's the parents are the ones that are most important. They don't have a 7.
Right.
Neither of the parents have a 7. So one possibility would have been that maybe he was homozygous and he was a 7 and a 12, and the test missed one of them.
Right.
But
That isn't
which we might not have known if we hadn't seen his parents. But now seeing his parents, unless
and the
there's
rest
some
of his
weird,
family.
correct, we now can't say that's true. So I guess that was helpful for me because I was able to consult somebody who I think would know as well as anybody. And again, his only explanation would be error. So
yeah. So all right. So I gotta say, whether it's Dan, you, whoever, whatever it is, what I'm taking away from this, and I'm not the only one because there's people in the chat saying the same damn thing, is DNA seems awfully less reliable than I previously thought it would be or was.
well,
it sounds to me like there's a fair amount of wiggle room.
There is. Okay. Can you see my screen? Okay.
Well, okay. Wanted to say what I was going to say though, but all right.
Oh, go. Okay, I'm sorry. Go on. Then finish what you're saying.
So my concern here is with this, what your friend Dan and you are saying could be errors, could be somebody could be entering the wrong thing, therefore being clerical error, all these various. This fills me with dread about DNA evidence and stuff, because this just seems like something that leaves the door open for the states, the prosecution in any given case, to be able to use it if they want. If it favors them, they're obviously going to want to use it. If it favors them to talk about how sometimes DNA can be wrong, or certain errors can whatever, to help them discount potential DNA evidence to help exonerate somebody and stuff like that. For me, this leaves a door open where the state can kind of use it both ways, to help them when they want, and to basically to say it's good when they want it to be good, and say it's bad when they want it to be bad. And that's what I don't like here. That's what that's the doorway that I see being left open with this.
Well, actually, I kind of see it the opposite. Actually, it's the prosecution and the forensics community that has been, and the courts that have been, up until recently, presenting DNA evidence as infallible, as irrefutable, as it's actually.
Well, that's what I'm saying. It's not, and that's the concern here for me.
Right. But that means that defendants now, or people, it doesn't mean we should throw it all out. It's not all bad, but it's not perfect. And there's different kinds of errors. And it's understanding how it works, and understanding what the errors are. It makes it in my mind, this is actually good that this is being recognized. And most of the people bringing this up are the people like this article I'm going to show you is by, if you've been, following, for the Innocence Project and DNA issues in the Innocence Project, then you'll probably be familiar with Greg Hampikian, who's a big name. He's a professor at Boise State, and he wrote this editorial in the New York Times because the NIST, which is the National Institute of Standards and Technology, the government basically actually did their own study. And this was a different type of error. But yes, this should give us pause, because if you go to my screen, he wrote an editorial to because this study was kind of tucked away. First of all, they did this study, and then they didn't publish it for a long time. And then, even as he claims in his editorial, the authors of this actually very enlightening study kind of poo-pooed their results. So is this on the screen? The danger.
Yes,
So this was in the New York Times, September 20th, 2018. The dangers of DNA testing. In a new study, and again, Greg Hampikian works. He's a professor at Boise State University, but he's also very active in the Innocence Project throughout the country and actually nationwide. And he's got a really good talk that, well, those of you who were in Scooby School, which is sort of in the process of being reconstructed, this is one of the first introductory lectures that I have people listen to, is one from Dr. Hampikian. So they get NIST. So this was done by the government. So if the government's claiming, that there's problems, says a lot to me. So what they did was they gave 108 crime laboratories, I don't want to say data. I mean, it wasn't a real crime. It was manufactured data, but they didn't know it, and they were told to analyze this. Now, this had to do with this is a huge problem area right now. Are these mixed samples and mixed profiles, and this is something that's becoming more of a problem, because it's kind of like with everything good, there also comes a negative. So as DNA testing becomes more and more sensitive,
Mm-hmm.
it becomes more prone to picking up whatever you want to call contamination or extra DNA. So it becomes more prone to error. So now they gave this crime, and in the study, 74 out of 108 crime laboratories implicated an innocent person in a hypothetical bank robbery. That's what? Like a 68% error rate. That's crappy. So whether you like it or not, we as a public need to be concerned. So he goes on, before you give police a DNA sample, read an alarming new study of crime laboratories published this summer. This is Hampikian, who again is one of, along with Dan Crane, one of the people that I consider the gurus. While I don't worship anybody, but I don't consider researchers, blah, blah, blah. The first two subjects, the test results are troubling, especially since errors also occur in actual casework. And then they talk about, a case, where Mr. Jackson spent nearly 4 years in a Nevada prison until the crime lab realized it had accidentally switched his sample with another suspect's tube. Again, another kind of error. But those happen. Switching tubes, mislabeling things. That's another kind of error. Tube swaps are easy to understand, but some laboratory errors are far more difficult to understand. For example, it's hard to interpret DNA mixtures from 3 or more people. As DNA testing has become more sensitive, most laboratories are now able to produce profiles from anyone who may have lightly touched an object. And so again, while some may consider that a good thing, it also makes things more complicated, because now you may be picking up DNA from people who didn't have anything to do with crimes.
Right.
Anyway, the other error, and this, I will have, a lot more to say about, and I'm talking about Sherry's, some of Sherry's work. Not only were there errors in in whether or not they implicated a suspect, but there were also errors, shocking, he called it. It was at how the labs differed in how they calculated their statistics. And statistics, is a whole other topic for another video. But the match statistics varied over a 100 trillion-fold range. And sometimes these numbers get so big that they don't, I think to most of us, they don't really mean anything. But I like how he says, that's the difference between soda change and the United States, gross domestic product.
Right.
So not only whether labs are, calling they're saying someone could be included, But then there's also errors in how statistics are used, and that's a big problem. And I have a great video if we get to it. I have lots of homework for you guys, by the way. Nobody's in the mood seriously, but it's good stuff. Anyway, so you can go to this. I'm going to leave links for all of this, but this is just one. With tens of thousands of DNA mixtures analyzed each year, there are many innocent people who hope crime labs and courts take the National Institute's study seriously and act quickly. I don't know, Erekose. You know what? When you told me, I said, I think the most likely explanation for the blood vial being different is error. And you said, I don't agree with that. I said, good. Good. Because
I
it's
don't
good.
necessarily agree, or at least I'm not
No.
as quick to explain it away as a typo or an error. I'm sick of those two things being used to exonerate the state in this. It's just beyond the point of being sickening at this point. But I understand.
Oh well, you should know, of all people, I'm in
I know.
no mood to exonerate the state. However, this wasn't the state, actually. This was LabCorp who made the error, actually. So anyway,
yeah.
However, now, and I will mention, I did have a long discussion today with my good friend Seeking Truth for Good.
Yeah.
Is her Reddit name, because that's what she prefers to go by. And she, because I'm not even going to get into this, however, she has proposed, along with some other people, with some good evidence, and I actually think it's a rabbit hole worth going down. That there's some issues, and again, this is kind of her thing, but she has a post on Reddit where she talks about blood drawn from Gregory Allen and the potential that maybe they sent the wrong blood to this LabCorp.
Oh, wow.
And so again, I'll refer you to, I don't know if she's even here on the chat. She might be in bed. She doesn't like to, she likes to lay low. But she said to refer people to her Reddit post. We talked for a long time, and I agree. That's compelling. I don't know enough about the details, but apparently there was blood drawn at the same time by Gregory Allen. For those of you not familiar, I'm talking about the rape case.
The guy who actually raped Penny Bernstein.
The guy who actually raped Penny Bernstein. Apparently, at the time that Stephen Avery's blood was drawn, 5 tubes of blood, which 5 tubes, that's a lot of blood. I don't know what they were doing with all that blood, unless he was having some medical testing done. I don't know. But one of those vials was sent to North Carolina. She said, okay, now what's in North Carolina? LabCorp, the place that tested these tests on Stephen Avery. So a vial, now she doesn't know where in North Carolina, but the fact that LabCorp was doing testing, for the nation at this time. Not very many state crime labs were doing this testing, so very likely his blood could have been sent to the same place. Now, again, I can't comment.
Though, is pointing out that the problem is the blood vial that was sent in for testing is said to have been coming from Kettle Moraine, and he was not in that prison in 1996. He was in Fox Lake in 96.
Right. So there are a lot of and these are the things that these sleuthers, are much better acquainted with. However, they've been talking to me about, and I agree. So again, which is why I say, barring any foul play, yes, error would be the most likely. However, I'm not saying foul play. Do you understand what I'm saying?
Yeah,
I'm not saying that foul play is not so right. Just like Dan Crane said also, there's no reason a 7 should be a 12.
Right.
Either it's a really crappy gel, either it's was a really crappy gel, or somebody made a typo, but now again, like I said, if I didn't know anything.
5 tandem repeats, though. I mean, short tandem repeats. Though. I mean, that's seems like a significant number to me. So.
Exactly, and usually the errors are closer. They'll like mistake.
Like a 9.3, which is essentially like a 9.75, really. You know, being
Right.
mistaken as a 10. Okay, I get it. That I can understand. That makes a little sense.
Right.
So, yeah.
So this is a good, but I understand your concern, but seriously, I think that the all of the errors coming out is only going to benefit it's not going to benefit the state. It's going to benefit the defendants who for all of these years have not been, people have pled guilty. Is it pled or pleaded? You know, I always get weird on those words. Pled. That's what I think it sounds right. But pled guilty because when they were innocent, because somebody said, oh, there's this DNA evidence we have on you. And they think, oh my gosh, I'm never going to be able to fight the DNA. This is giving people who are wrongfully convicted more power because there's evidence that yes, and
Yeah.
no, yes, they are rare, but errors.
It may cut both ways, but somehow I see this little discrepancy favoring the prosecution more than it favors anybody else. They'll ignore
Okay.
it when it favors when it suits them, and they will use it when they are trying to discount somebody's DNA evidence. So
But it's
that's
but it's but it's so much biased in favor of the prosecution right now. That I mean, look at
everything is exactly.
the prosecution has access to labs, has access to testing. I mean, look at Stephen Avery with his, you know, $400,000 seems like a lot of money, but it wasn't enough money to get them a decent DNA expert. You know, to even counter any of the you know, any of the evidence in that trial, which most of should have been tossed out. This is a good website I'll put that people should look into, that get hooked up with the Council for Responsible Genetics, GeneWatch. This has a lot of articles about what's going on in the state of forensic testing. And here's an article, the potential for error in forensic DNA testing. But this promoters of forensic DNA testing have from the beginning claimed that DNA tests are virtually infallible. In advertising, materials, publications, courtroom testimony, the claim has been made that DNA tests produce either the right result or no result. This rhetoric of infallibility took hold early in appellate court opinions, which often parroted promotional hyperbole. That's right. It was supported when the National Research Council, in the second of two reports on forensic DNA testing, declared the reliability and validity of properly collected and analyzed DNA data should not be in doubt. Blah, blah, blah. Wrongfully convicted people were shown, okay, it was further, blah, blah, blah. Anyway, the rhetoric of infallibility proved helpful in establishing the admissibility of forensic DNA tests and persuading judges and jurors of its epistemic authority. Anyway, you know, I understand your concerns. However, this atmosphere, or what will you, that was set up is what allowed the forensic community to all of a sudden, how can I say, sort of abuse the science and take things now to allow a profile, sorry to rip on Sherry, but to allow a profile like BZ, which never should to be admitted into court, to allow them to take more and more leeways away from what is considered the proper use of this data without any accountability. And that has created, you know, anyway, in this article, I will argue that this shared assumption is wrong, although generally quite reliable, particularly compared to other forms of evidence used in criminal trials. DNA tests are not now and never have been infallible. Errors in DNA testing occur regularly. DNA evidence has caused false incriminations and false convictions and will continue to do so. Although DNA tests incriminate the correct person, in the great majority of cases, the risk of false incriminations is high enough to deserve serious considerations in debates about the expansion of DNA databases. And then that's a whole, there's a couple of other issues that I want to talk about, probably in other videos, because you're falling asleep on me. And Dr. Pepper isn't going to do it, and I know you have to work, because there's two specific things that I want to talk about that they're doing, but probably in part 2 of this. And one is familial testing, which is a big thing now. They're not going into databases and just looking for matches. They're going into the database and they're looking for someone related to you that maybe they can pluck you out. And there's some serious problems with that when we look at these STR loci and what can happen. So short tandem repeats, we think of them only as these forensic, in terms of these forensic things, but a repeat, that just means like a repeating unit of DNA. These are all over the place, okay, in our DNA. I think one article I read said we have like 20,000 of them, and they're not really understood. But or science is still in the process of understanding the genetics of these, the inheritance of these. One of the reasons they're so useful in discriminating between people is because they have a high mutation rate. And I've actually read literature from the forensics and FBI sources that say, oh, these are very stable locations on the chromosomes. They have a very low mutation rate. Well, that's just bullshit. They have a very high mutation rate. And so having a very high mutation rate, there have now been shown, and I have another study to show you, that there are cases, especially with men, where there can be a mutation between. So mother and father should have, mom has, I'm getting ahead of myself. Mom has, you get one chromosome from mom and one chromosome from dad. So if mom is 14 and 11, okay, and dad is 8 and 10. Okay, then,
Right, you get one of each. Right.
Son could be 14 and 10. You have to have one from another. Okay.
Right.
Well, it has
and
been.
It's possible to get the same one from each and have only one.
Correct. But it has been shown in a statistically significant way that because of the high, the cases where usually from the father, there's a mutation. So son could show up like that.
Okay.
And it's not common, but it's not rare. I'll have to pull up the studying the statistics.
Right.
Because there can be mutations, and this is the kind of mutation, this is called a germline mutation. So this is a germ being in the sperm or the egg. So now think about this. If you're going to do if this exists, now let's get the study. If this exists and you're going to start doing familial testing, then that's going to be a problem. Oh, this is what I wanted to show you. Okay. Can you go on? Then that's going to be a problem when you are starting to try to identify people by who they're related to. If you were this person, then, and I started looking for relatives of this person, I'm going to get the wrong relatives because this person had a mutation. Again, it's probably less than 1%, but when you're talking about 1% of a database of 13 million, that's a significant amount of people.
Yes.
So these are things that need to be considered. Okay, I'll just show you one more thing, and then we'll stop or if there's any questions or.
I was going to say, let's leave the rest of the more technical stuff, obviously, for next time. I think we've given everybody enough to think about. And everybody is very interested in looking into the statistics, the study on the New York Times article that you posted earlier.
Yeah,
So
yeah. I'm
that's,
gonna leave.
you know, so we've already given people, you know, some stuff to think about. So
Okay, can I just show you one
sure.
more thing? Okay, because this is so this is what I went when you talked about, you know, the test being crappy. So this is okay. This is NIST, and I'll tell you about this website. So what they've done um, is they went and they looked at they decided to see, you know, how accurate to check up on the accuracy of the testing. And this was in 2017. They updated because the population, you know, when you look at the statistics, they're all based on how many people have this allele and that allele. So they took they went back, and I'm just going to look. This is, I don't know if you can see this on my screen. I'm pulling it up, but here's the table. They took 1,036. So out of this database, the last number I heard was that they have 13 million people in the U.S. database. Okay,
Mhm.
they took out 1,036. Okay, so a very small sample size.
Right.
Okay, they retested them all to see how accurate they were. Okay, they found 12 that were wrong. Now you can say 12 out of 10,
12 out of 1,000.
1,036
Right.
is a pretty, you know, that's not a lot, but it's not insignificant either.
Right.
Again, out of a database of 1 million people. So let's look at the first one here. Sample number 1, African American. And then the sample has a name. It has a code number. The locus is D5. The original profile was 1212. The revised profile 712. Kind of ironic. I mean, it's just ironic that it's a 712 switch.
Yeah.
Not unheard. And this wasn't with the crappy old electrophoresis gel testing. This was with the newer testing. Again, you know, it's not common. Most of them, if you look at, you know, 2020 to an 1820, 1515 to a 1315, 11, you know, so most of them are very close. But they were wrong. So now they updated. The article talks about how they updated the database, and this is very small. But again,
Right.
this
So
was
what's
just
interesting here is you're actually illustrating where they actually show where DNA was retested and a 12 became a 7.
yeah.
In the case of what we're talking about here, in this case, we're talking about a 7 becoming a 12, which is a little different, but you know, you know, I guess it's noteworthy.
Why is it
It's
different? Why is it different? Why is it different?
Well, because
It's still,
in this case that you're showing here, a 12 becomes a 7. But
yeah.
in the case of what we're talking about with Avery, a 7 is becoming a 12. It's going in the opposite direction. You see what I mean?
Right.
Instead of a 12
But
becoming a 7, it's a 7 becomes a 12. But you know,
okay.
anyway.
Well, I guess what was more significant to me was
But
the
it's the fact
number,
that it's the
the
same number,
spread.
and it's a discrepancy, right?
Well, it's the same that there were there are 5 base pairs or 5 repeat. I'm sorry, 5 repeat pairs apart. So, again, I'm not trying to make. I'm trying to just present, as always, just present the information. I'm not. I believe in, you know, people should, you know, be informed and critically think about these things, and like, yeah, it's not uh, it's not there's no easy answers, there's no easy,
Well, it's DNA. It's complicated, right?
it's complicated, and it's
So
late,
hey,
and we'll
it comes down to collection methods. It comes down to straight up lab tech error. I mean, look at Sherry Culhane herself. She's the fastest there, but she makes the most mistakes as well. So
well, fast isn't yeah. Well, yeah, there could be something to, yeah, fast does not necessarily mean more accurate, and which is another concern, because that's what most that's what I hear about, you know, from most states and government agencies. They got these backlogs. They want more data processed faster. And by
right.
the way, that's one of the criticisms of So now we have more new technology coming. Look, we're still trying to figure out this technology. Now we have things like, rapid DNA testing coming in. And one of the criticisms of companies like, at least with these, so I can go like to, I can go on to the NIST STR database, and I can look at, okay, so this is THO1. This is the loci, locus is singular, loci is plural. Locus THO1, I can look up all of the primers that are used. So PowerPlex. So Wisconsin State Crime Lab uses the, not this one, PowerPlex. They didn't use a 1.1. They use a PowerPlex.
Well, we'll leave all that
16.
for next time. That's
okay. But anyway, what I'm saying is it's all published. But now, like Andy, they don't publish their information. It's all proprietary. So some people are concerned about that because now a defense attorney can't go and say, "Ooh, maybe your primers were wrong," and because that's another
that'll be
issue.
a problem moving forward. That'll have to be obviously.
Yeah,
there needs to be transparency. That's for me. That's a huge part of our legal system. There needs to be transparency.
Right, and so now, So anyway, for people, I do apologize for the lateness and the delays. I've had some family medical emergencies and sicknesses that have caused me to be not available and did cause my delay. So I really do apologize, and I appreciate the people that are here. And I don't know. I think this is hopefully I'll be working. I certainly will be talking to my fellow sleuthers and exploring this blood situation with the other aspects that they have done more work on with as far as researching what happened with this blood, etc. And the topic of the errors in the DNA, if people want to hear more about it, is obviously interesting to me. And then I have my mitochondrial DNA presentation waiting for you.
Yeah.
Which I know will probably keep you up at night.
Oh, yes. All right.
Okay.
Well, it's been fun, obviously.
Go get some sleep.
Hit the high points on this a bit, and so we'll come back and get a little deeper in next time we come back and revisit this. Obviously, we can revisit it as part of Scooby School when we start doing that or something,
So,
whatever we do.
The bottom line answer is, why was the blood vial different? I don't know.
Could be nefarious means. Could be,
it could have been. It was a really
or user error. So, right? All right.
But from the perspective of the testing, the most logical. Now, again, when I talked to Dr. Crane about this, I did not fill him in on any of the other background. I just showed him, the results. So again, given that, but again, we don't know. I've certainly seen the raw data. In this case, a gel likely, which looks like. I was going to show you a picture. Okay, yeah. Here's a picture of what Okay, this is a picture of what these gels look like, as opposed to those, the peaks from the electrophoresis. So what they get, they run the DNA down on a gel, and it's labeled with a radioactive tag, and so they get these bands.
Right.
And they're looking at the darkness of the band. So it's a very, you can see if it's blurry. Right. It's a different
type of thing, right?
Different type of thing. So what he was suggesting,
right?
What he was suggesting was that possibly it was like a really crappy gel, and maybe they just couldn't tell, and wanting to get results, they just said, "Oh, this one looks good."
Right. Absolutely.
But we don't. Again, we don't know. Okay.
Right. Okay, guys, that's about it for tonight. Hope you guys found it interesting. If you haven't already, please hit subscribe, and we'll see you.